In this video, we'll take a look at molecular genetics, which is what my own training and research is in, so this is the subdiscipline of genetics that I'm the most familiar with. As its name suggests, molecular genetics studies the molecules of the central dogma and how they control traits. And in previous videos, you've already seen what these molecules are in the central dogma. They're DNA, RNA, and protein. So molecular genetics is the study of DNA, which we'll study in chapters 10 and 11, RNA, which is chapter 14, and protein, chapter 15. And it's the study of the processes that make these molecules, namely DNA replication, which we'll cover in chapter 12, transcription, which is chapter 13, and translation, which is chapter 15. Another way that we can define molecular genetics is that basically it's the study of gene function. In our example, this gene's function is in melanin production. But how exactly the protein encoded by the gene carries this out and how this gene is regulated is of primary concern to the field of molecular genetics. And this is actually pretty close to what I would tell my parents when they used to ask me what I did for a living. I would just say, I figure out what genes do and how they do it. Molecular genetics is also the study of gene regulation. How a gene is regulated determines how much protein that gene produces at any given time. For example, when our skin cells are exposed to sun, genes encoding melanin-producing enzymes increase their expression levels, causing more melanin to be produced. So we would say that this gene is regulated, in this case activated, by sunlight. And we'll look at gene regulation in chapter 16. Now there are many different methods to determine the function of genes. One approach is to determine the DNA sequence of a gene, and then use the genetic code, which we know, to determine, one codon at a time, the amino acid sequence of the protein that gene encodes. If we align the amino acid sequence of the protein that our gene encodes with the amino acid sequences of other proteins, we can see which ones have a very similar amino acid sequence to ours. And if they do, we can guess that our protein probably has a very similar function, and design experiments to test that hypothesis. There are plenty of other techniques out there for studying gene function, and we'll talk about a few of them in the next video on model organisms. Keep in mind that these three subdisciplines of genetics that we've discussed can and do overlap. For example, cancer genetics often involves determining the function of genes that regulate cell division, and whether cancer-causing mutant alleles are passed from parents to offspring, and how common these alleles are in the human population. These subdisciplines are only one way to divide the field of genetics. There are others, like dividing the field by organism. For example, I consider myself a plant geneticist, while others may consider themselves part of evolutionary genetics, human genetics, or even microbial genetics, and these are just a few. It's a large and diverse field, and it's a lot of fun to work in. Okay, so at this point you should be able to explain what each of the three main subdisciplines of genetics studies, as well as a few of the methods that each subdiscipline uses in their research. Now, as usual, feel free to rewind and watch the videos again if you'd like to review. Now, in the next video, we'll look a little more at genetics research and the essential role that model organisms play in determining the function of genes. See you all later.